This next section focuses on the difference between DNA and RNA. So we're going to fill out this table. First of all, the name is different. So for DNA, it's deoxyribose. For RNA, it's ribonucleic acid, sorry. For RNA, it's ribonucleic acid. And this difference comes based on the fact that there's a different sugar. So for DNA, it's deoxyribose sugar. And for RNA, it's ribose sugar. Basically, the sugar in DNA is missing an oxygen versus the sugar in RNA. Number of strands. Typically, DNA is double-stranded. However, there are exceptions to this, even within eukaryotic cells, but even especially in like viruses. RNA is typically single-stranded. And again, there's always exceptions to this, but this is just the general, most common scenario. As for the bases, so I said that the bases of DNA are A, C, G, and T. Adenine, cytosine, guanine, and thymine. In RNA, we actually replace thymine with uracil. So our bases are A, C, G, and U for uracil. Uracil is also considered a pyrimidine. Types and functions. As far as DNA goes, we have our genomic DNA, which has the function of storing all of the genetic material in the cell. This constitutes all of the instructions for making an individual unique. Additionally, in eukaryotic cells, we have mitochondrial DNA. Mitochondrial DNA is kind of specific to its bacterial origin, but there are some genes on mitochondrial DNA that have been associated with human disease. Everyone is 100% matched with their mother for mitochondrial DNA. If you think about how an individual is created, you have a sperm cell and an egg cell, and they fuse and unite through fertilization and then start to divide to create the individual. The egg has all of the organelles. The sperm usually just has one terminal mitochondrion, which is completely destroyed by the time the sperm penetrates the egg. So all of the mitochondrial DNA from the sperm is usually destroyed through that fertilization event. So we get all of our mitochondrial DNA from our mom. RNA, there are many types, and we won't get into all of the types like siRNA, mRNA, or miRNA, and all those types of microRNAs and double-stranded RNAs, but we're going to talk about the three main types. rRNA is ribosomal, and that helps make up the ribosome. tRNA is transfer, which helps transfer an amino acid to a growing chain of polypeptides, or growing polypeptide chain in translation. And then we have mRNA, which is for messenger. Messenger RNA is used in transcription and translation. So basically, it's an RNA copy of a gene of interest, which can be read by the ribosome to create a protein. As far as locations go, in a eukaryotic cell, DNA is found in the nucleus. In a prokaryotic cell, it's going to be found in the nucleoid. which is basically just a region where the DNA piles up. RNA can be found in numerous locations inside of the cell. It can be in the nucleus, it can be floating in the cytosol, associated with the ribosome, so we find RNA in multiple locations.